Hi there, we're back today and it's a very exciting day. We are going to dilute our genomic DNA so that we can send our isolated strains for sequencing. Um, so we should have already gotten the concentration of the genomic DNA through a NanoDrop, which tells us both the concentration and the quality of the genomic DNA extraction. And so generally I'll do that by documenting the nanograms per microliter as the concentration and the 260 to 280 value as a purity check, let's say. Now from there we have, I was able to isolate 8 different strains, of which we are going to select 6 of those to send for whole genome sequencing. Now we've identified strains 1 through 4, and then 5 and 8 as the strains that we're interested in sequencing. And we should have done a calculation to get the strains to be greater than 10 nanograms per microliter and to be in 40 microliters of solution. And so what we're looking at is a concentration volume calculation, which we should do, and I should have a picture of an analysis of that available so that you can see how to do that. But then also now we need to do the actual dilution. And so the easiest way to do this is to do the calculations and then put that into a table so that it's easier for you to see when you're actually doing the bench work. The way that I like to do this is to label a table where it's your strain and then the amount of volume you need of your sample. This would be of the genomic DNA, and then how much water you would need to add. If you want to get really crafty, and things that I've done previously but I didn't leave space for here, is that if you wanted to make a bigger table that you only write out your strains one time, and then you have the concentration right there, the volume right there, and then you can add extra columns that would also include this information so that it's all in one space. That doesn't always happen when you're writing in a lab notebook, and sometimes you get better ideas after you've already done it, and so that's absolutely okay. It happens all the time. So what I've gone through is calculated how much of the sample and how much water I need. I would generally recommend always starting with pipetting the water first. This ensures that you're not going to get contaminated because once you start working with the DNA, you want to be very, very careful that you're always getting rid of each pipette tip every single time. Another reason that I want to pipette the water first, especially in this situation, is that I'm pipetting quite a bit more water than I am of the genomic DNA. So generally you want to pipette smaller volumes into larger volumes. Okay, so the first thing that we're going to do is we are sending our samples for sequencing in microcentrifuge tubes, and they have screw caps just to make sure that the lid doesn't pop open while they're in transit. And I've gone ahead and labeled everything on the side and on the top. You can see what we got there. Yeah, PL1. Cool. So the first thing that we'll do is we'll take our pipette. The first volume of water that we're pipetting is 31.29. So we need a pipette that can do that. I have a 100 microliter pipette here, and we're going to go 31.2 is going to be the closest that we can truly get to this. 31.2. I will show you 31.2. And we are going to take the water here, draw it up. Unscrew the lid, and then watch the water go into the bottom of the tube there. Now, because we're pipetting water into empty tubes, and you're doing this first, you could just use the same pipette tip as you go along. But if you want to be really careful, as I do in this case, because we don't want anything extra to get into our sample, we're just going to do the same thing. Take a new pipette tip. Our next amount of water is 32 point, let's say 4. So not too far of a movement there, 32.4. You can see that I get to the 0.4 by going a little past the 0.2, and there are little ticks there on the bottom that indicate what the points would be otherwise. Okay, so something I want to note right here is that before I pipette this water, because I think that this is a good moment to talk about this, the way that I know which sample I'm working on in a brainless world is when I'm done with a sample, I move it one over, I move it one up, I just move it so that it's not in line with the rest of them. So that if I look down and I say, oh shoot, I forgot my place, where am I gonna go? I know that this one was already moved over, so I'm gonna work on this one now. All right, so instead of pipetting all this water, I'm just gonna go over what we're going to do for pipetting the DNA now. So for the DNA volume, I have 8.71, and I'm gonna use a smaller pipette for that. I'm gonna use a 10 microliter, and I've already taken out my DNA samples and put them on ice. So I have tubes here, they're in the same order, and they're already labeled from before. And I'm going to shake it up a little bit by just like flicking the tube on the bottom. Make sure we get everything up there. And now with a 10 microliter pipette tip, double check that it's the right one into the correct sample. So we're gonna go, the PL1 is the first one. I'm going to adjust the pipette to be 8.71. And in this case, the tick marks indicate the point value here, 8.7, and then the smaller ticks would be my 7.1. We're gonna pipette mix this. So pull it up and down multiple times. And so into a tube containing water, we're going to add the concentrated DNA. And now the concentration of this tube should be 20 nanograms per microliter in 40 microliters. And it's going to go and be sent out for sequencing. So stay tuned. We'll see what happens next.